material that's more related to oxidation reduction. In particular, today's lecture will be on some aspects of the nitrogen cycle, which is a little bit of a diversion. It doesn't quite connect up with everything else we're doing, but we'll hit some principles that are going to be important in learning about oxidative phosphorylation and photosynthesis. And then also the material is really cool because it's just so fundamental. So we're going to talk about how nitrogen is cycled in the biogeosphere, which is really an aspect of a field of science which you could think of as almost think of as Earth system science, right? The cycling of the elements, the evolution of the Earth. We talk about this a little bit with carbon already because we talk about fixing carbon and then we talk about respiring carbon. So that's part of the carbon cycle, CO2 in the atmosphere, organic molecules on the surface. The planet is putting its carbon in different places. Some of it way underground as fossil fuels, which we're now digging up at a rapid rate. Some of it in the biosphere on the surface, and then some of it in the atmosphere as CO2. So similarly, nitrogen is in the atmosphere as N2 and is on the planet's surface as fixed nitrogen in different kinds of oxidation states. The most reduced of which is nitrogen, is ammonia, right, NH3. So, we're going to look at this reaction. This is a reaction that occurs when we do it industrially. So in sometime around 1905, something happened in Germany that was pretty transformative for how human civilization developed in the 20th century. And that was that the German chemists developed this process industrially, which is called the Haber process after the guy who invented it. And this is, of course, a gas, right? This is a gas. And so we're fixing ammonia now. This ammonia is now going to be fixed in the aqueous form. And, we could also write, of course, it'll protonate. It could become ammonium ion, right? And so this is called nitrogen fixing. And the fact that we can do it now industrially means that we can make a huge amount of this. And this stuff is basically fertilizer. Right? So ammonium salts, ammonium nitrate, and phosphate salts are the primary nutritive ingredients in fertilizers that crops need to grow. And so you could ask, well, where do these components come from? Right, in our industry today. So this guy comes from air, right, which is 79% by volume nitrogen, molecular nitrogen, and generally inert, right? You put something under nitrogen, you preserve it. You have an old book in the library, you want to keep it pristine. You don't leave it in the air where there's a lot of oxygen to start running oxidative reactions. You put it under N2 so that nothing happens to it. So consider that when you think about this reaction. If you're going to combine N2 with hydrogen, it's going to be pretty tough to react for the same reason, because N2 is unreactive, and that's because this is, of course, a triple bond, and so therefore highly unreactive. So even though this process is favorable by something like minus 30 kilojoules per mole, we'll write the number later, kinetically, it's really tough because there's going to be this massive activation barrier associated with breaking the triple bond. Right? So this comes from here, and even recently you could find people in the chemical industry talking about the miraculous nature of this reaction, because the components of it, they just come from thin air. Just amazing. We can just get all this free food for the crops that we need from thin air, right? Of course, that neglects where this comes from, right? So where does that come from? Anybody have an idea? Where do we get molecular hydrogen, which of course is fuel, right? You can burn it in oxygen. It doesn't produce any CO2. Well, you can electrolyze water
Yes.
and put in energy for that and get some H2 out of that, but that wouldn't. That's not done on a scale nearly large enough to generate the H2 for this. Yeah. So renewably, you could imagine using renewably generated electricity, like wind or solar power, to provide electrons to drive that cell, right, to generate H2 from water. And so in the long run, that's a good answer. Where do we get it now?
From air gas.
From where?
Air gas. From
Air
the company.
gas? From the company, from Air Liquide? Good answer. Where do they get it?
They pressurize it somehow.
This reminds me of the Charlie Brown comic strip. Why do they have a lot of rain in Oregon? Because there's a lot of clouds. Good answer, Charlie. Right? Where is this H2 coming from?
Could we maybe just oxidize methane?
Well, H2 is already very reduced. You oxidize methane, you're going to put some heteroatom in it, so you're not going to get it from that. But methane is, in fact, the source as a reduced hydrocarbon coming from fossil fuels. Methane and other fossil fuels, through cracking processes, through other refinement processes, eventually will generate H2. So you're right. It doesn't come from oxidizing, but it does come from fossil fuels. CH4, various oils, etc. Right? So, in order to extract in order to get H2 gas out of the fossil fuels, of course, fossil fuels have carbon in them, and so we're going to be putting up a lot of carbon dioxide. So, of course, this process is very polluting in the sense that CO2 is a pollutant and driving up global temperatures by trapping heat, by capturing IR that's re-radiated from the Earth and sending some of it back into the Earth. But this process to make the ammonia from fossil fuel derived hydrogen is the reason why we can get all this fertilizer, right? And so in the year 1905 or so, the population was about 1.2 billion, something like that. And in 2016, the population is about 7.3 billion. So, we got plus 6 billion out of this transformation. There's no way that we could support the current population of the Earth without, the agricultural practices that we have. Right? So we could consider that we have used this reaction to upset the nitrogen cycle, and the numbers. You could look at this if you take a course in this sort of stuff. It's quite interesting to look at what are the numbers in the nitrogen cycle without anthropogenic interference, without what humans do, and what are the numbers now in terms of how much nitrogen is in different places and what are the fluxes. And we have huge effects on the nitrogen cycle by virtue of just having so much population and fixing so much nitrogen, and that's driving other aspects of the cycle to be different from how they would be in nature without us. But also, we have a practical problem to solve here. Fossil fuels are, not infinite, right? They're a limited resource, of course. Not to mention that there are climate effects from burning them, but they're also just limited. So we're going to have to go to something like what you suggested if we're going to continue to make the hydrogen to create enough ammonia to grow enough food. Right? So that's kind of an important impact of this stuff on where we are now. But let's look at how it's done in nature. And this process is done in nature, right? This is going to take, I don't know what, let's say 600 degrees C and 100 atmospheres, or something worse than that, right? In industry, that's what we do. So in nature, this is done by an enzyme called nitrogenase. So this is a picture of the nitrogen cycle from the book. And so nitrogen fixation is the most important process to make nitrogen biologically available. There's also other pathways here. You can see that there's this pathway here. We're not going to talk about it. It's an anaerobic pathway. It occurs in archaea, which are microorganisms in the third domain of life. So there's eukaryotes, there's bacteria, and then there's archaea. It used to be eukaryotes and prokaryotes. now we recognize from all the sequences we have that the prokaryotic domain of single-celled organisms is in fact, at the molecular level, really understood best as a bacterial domain and an archaeal domain. Right? And so that's the archaeal domain is where this Animox reaction goes, and it's a way of generating ammonia from nitrite. But this is still the most important process here. So this is done by nitrogen-fixing bacteria in archaea. It's a process that's very oxygen sensitive because it involves a lot of redox cofactors that are sensitive to oxygen that will react, become oxidized by oxygen, and then be unavailable to participate in the process. And we'll look at what some of those are. Right. We looked at this a little bit here, right? Amino acids in the diet are being thrown away in recycling proteins. Eventually going to go to ammonia, right? So they're excreted. Ammonia has to come out of say vertebrate organisms in various ways, either as ammonia or as urea or as uric acid. But those products would break down to ammonia in the environment soon enough anyway, or else they'd get processed into oxidized forms, which would enter the cycle one way or the other. So we have these different reactions that are going on. So one of them is nitrification, which takes ammonia to nitrite, and ultimately to nitrate. So we have the different oxidation states here of the nitrogen. We have 0, -3, +3, and +5. So we have a difference of 8 in the oxidation states. From -3 to +3 is a difference of 6. So think about this when you think of, what are the enzymes that carry out these transformations? What do they have to do? How many electrons do they need to provide to their substrates or take away from their substrates when they're catalyzing these transformations? Right? So nitrogen fixation, nitrification is oxidation of ammonium to nitrate. This actually serves as an energy source for some soil bacteria because this is energy rich. And then nitrate assimilation is going from nitrate back to nitrite to ammonia, and that's not actually shown on this slide. But I would say if you have this printed out, draw the lines back in because that's also an important aspect of things. In fact, the textbook talks about the enzymes that do this. It talks about nitrate reductase and nitrite reductase. It talks about those two enzymes, which I have on slides. But this pathway that's shown, actually the authors have apparently decided to keep everybody in the dark about what actually does those. So I thought in the interest of completeness, we should have a slide that shows kind of what's going on here. So this is -3. This goes to NO2 nitrite. So this is +3. And this goes to nitrate +5. Then there's this process here, nitrate getting assimilated back into N2. Okay. So then we also have this reaction and this reaction. So this enzyme here is nitrate reductase. Pretty easy to remember, going from nitrate to nitrite. And then this enzyme here is nitrite reductase. Going from nitrite to ammonia. Okay. And then here, these steps are the nitrifying steps. And so these can be energy yielding. And these are carried out by some enzymes that go through hydroxylamine as an intermediate. So here we've got -3. This is actually -1. This enzyme is called ammonia monooxygenase. It's an oxygenase, so it's actually taking from molecular oxygen and going to leaving an oxygen in the product. So this is hydroxylamine. And then this goes to nitrite. This enzyme is called hydroxylamine oxidoreductase, which is a multi-heme enzyme. Multi-heme, that's not all legible. And then to go from here to nitrate, this is called nitrite. You still on the page? Almost. Nitrite oxidoreductase. So it's just so you have, I won't ask you to remember these names, right, but of these enzymes, but it's just so you have some sense of what the process is, that there actually are enzymes associated with these processes to go from ammonia to nitrate as the textbook shows you. So ammonium monooxygenase, hydroxylamine oxidoreductase, nitrite oxidoreductase. These are complex multi-subunit redox enzymes that are going to function in ways that at least have some parallel to the nitrogenase that we're going to talk about in more detail, and the other enzymes, the nitrate reductase and the nitrite reductase that we'll at least mention briefly. Yeah.
So the Ford reactions are different. It's not just reversible.
That's right. These are not reversible reactions. These are all individual enzymes going on in different bacteria and archaea. Different organisms are conducting this process versus this process. And the enzymes are unique. This is one unique enzyme, another one, another one, another one, another one. That's 5 enzymes right there. And then this enzyme here is nitrogenase, which we'll talk about soon. Right, so that's 6 enzymes, and then there's more enzymes here. I don't know the names of those off the top of my head. It's all enzymatic. It's all in biology. So all of these transformations are possible. And then we also have the pathway where we have nitrite and ammonia coming together to form gaseous molecular nitrogen. And there's a couple pages in the text, in one of those boxes in the text, that tells you more about that process if you want to read about it. Very new. We don't know a whole lot about it yet. Although there are lots of gaps in our knowledge of all of these enzymes, really. Because when we get to these more complex redox reactions, and we'll see this again when we talk about oxidative phosphorylation in the mitochondria next week, there's a lot of details about this process that we don't understand yet. Yeah.
So are there bacteria that we can grow to do these processes?
That we can grow in the lab?
Yeah.
I'm not familiar exactly with which bacteria are culturable and studyable, but a lot of them probably are, or at least some of them are. I'd be surprised if we had no ability to culture any of these. We find them in the environment, and then the process in microbiology, one of the big things to do, and there's a number of labs here at PSU who are doing this stuff, is to go out and find the bugs, go to the bottom of the Pacific Ocean or go wherever you need to go, and then bring them back to the lab and see if you can get them to grow in pure culture. And sometimes you can't get them to grow in pure culture, but you can get them to grow in syntrophy with another bug, that they provide mutually important metabolites for each other. So yeah, we would have to go and look into exactly which ones are amenable to experiments right now, which ones are not. Okay, so that's the overall nitrogen cycle. So here's the slide again. The red arrows are showing the processes that are going on that are anaerobic. Notice that these processes here involve actually adding oxygen in. So these are aerobic processes. There's O2 involved in those enzymes going from here to here. Okay, so if we look at nitrate assimilation. So nitrate assimilation then is the process that's not shown here, right? It's going to go from nitrate to this and to this. So we have these two enzymes, nitrate reductase